students, welcome to Library, and let's start your today's test, that is test number 2, and it contains questions from nucleic acids as well as fats, as I told you. Okay, so one thing is missing in your first slide, that is nucleic acid plus fats. So you have 113 question, out of which approximately your 70 to 70 questions I've made from biochemistry, that is nucleic acid part, and from fatty acids. Rest of the questions till 113 are from fats. So total question 113. Time limit is 2 hours. Marks is 3 mark per question. Yes, there is negative marking, that is 1 mark will be deducted for every wrong answer. So what's your target is you should score 185 plus. Okay, your BARC cutoff doesn't go very high, that is during my time it was 120 marks, and last to last year it was 1 to 2.5 marks. Okay, so it ranges between 40 to 45 questions, but then again, just because we are preparing for it, therefore we should be always ahead of the exam's real cutoff. Okay, so try scoring 185 plus. What are the instructions? The instructions, everything has been written on the preceding page, but the only thing is there are total 113 questions in this test, just to keep you ahead of time. Otherwise, how many questions do you have in reality? That is 100 question, and the time which is allotted is 2 hours. I'm trying to make you more competent by making you solve 113 question instead of 100 question. Okay, such that you at least have a virtual practice of solving those many questions. Okay, so let's start. First question says a nucleoside consists of, out of the 4 options, you can pause your video and then you can click, or you can write in your notebook that what is the correct answer. After 1 hour you will get the answer key for it. Okay, so the second question is a nucleotide consists of, again pause the video, read the options and take it for yourself or write it for yourself. Third question, a purine nucleotide is, out of these 4, again do the same thing. Okay, you just make a column of 113 questions and start writing the answers in your copy. Fourth question, a pyrimidine nucleotide is, okay fine, again you have to do the same thing. Fifth question, adenine is, that is the chemical name of adenine. Out of these 4 options will be, you have to write your answer in the copy. Then sixth question, 2,4-dioxypyrimidine, that then this chemical name is the like chemical name like we used to do during your organic chemistry. Okay, so this name signifies which of the nitrogenous base, thymine, cytosine, uracil or guanine. Write for yourself. Then seventh question, chemical name of guanine, you need to remember all of them, like all the chemical structures you have to remember. Eighth, nucleotides and nucleic acid concentration are often also expressed in terms of, very important and easy question, again write your answer. Ninth question, the pyrimidine nucleotide acting as the high energy intermediate is, again an easy question, write your answer. So don't think that BRC always asks you easy questions. It's a mixture of easy as well as difficult question as well as very easy questions, and it's a game of timing only. Okay, and also let me tell you that, see, many people are making videos regarding BARC exam, but it's your moral responsibility to call it BARC, not BARC, because see, BARC is also given, the term is also given for the barking of a dog. Therefore, the scientists never in BARC, they never prefer to call it that way. इसीलिए if wherever you hear the name bark, or if people are calling it, let's prepare for bark or something like that, just start thinking that, they are very naive, they haven't dig deeper into the topic. That is, let us not call, just do one thing, let's never call it bark. I always call it BARC. Lot of students have commented and asked what is BARC. BARC is a research organization, you do not need to call it bark. Okay, so never ever call it bark. Then the 10th question says the carbon of the pentose in ester linkage with the phosphated nucleotide, this is structure. Structure is, you have to point out the carbon of which is attached to the pentose. 11th, uracil and ribose form. What is the structure which is formed by this uracil and ribose? Again a very easy question. 12th question, the most abundant free nucleotide in mammalian cells. Easy but confusing question. 13th question, the mean intracellular concentration of ATP in mammalian cell is about. I'm giving these questions so as to make you more, competent, as well as to make you realize that yes, you can get some of the very straightforward and unexpected questions there. 14th question, the nucleic acid base found in mRNA but not in DNA. Very easy. 15th question, in RNA molecule caps, what is the function of the caps? Again, a very easy question. 16, in contrast to eukaryotic mRNA, prokaryotic mRNA. Easy question. 17th question, size of small stable RNA ranges from, out of these four options, write your answer. 18th question, the number of small stable RNAs per cell ranges from. This is easy, confusing, as well as thoughtful, I would say. If you haven't read it as such, then it will be thoughtful question. 19th, molecular weight of heterogeneous nuclear RNA is. Then 20th question, in RNA molecule, our time has exceeded. I had to upload this at 9. I'm recording at 9:11, so sorry. In RNA molecule, guanine content does not necessarily equal its cytosine content, nor does its adenine content necessarily equal its uracil content. Since it is again a very easy but applied question. 21, the nitrogenous base present in the RNA molecule is. 22, RNA does not contain. 23, the sugar moiety present in RNA is. You have to pause the video, okay? 24, in RNA molecule, just out of these four options, write your answer. 25, methylated purines and pyrimidines are characteristically present in. Again, write your answer. 26, thymine is present in. 27 is missing, so sorry. 28, in every cell, the number of tRNA molecule is at least. Again, thoughtful. 29, the structure of tRNA appears like. Very easy question. 30th question, although each specific tRNA differs from the others in its sequence of nucleotides, all tRNA molecule contain a base paired stem that terminates in the sequence CCA. So it's basically asking where CCA is located. Okay. So 31st question, the transfer RNAs are classified on the basis of the number of base pairs in dash. 31, again easy question. 32, in tRNA molecule, D arm is named for the presence of the base. Very easy question. 33, the acceptor arm in the tRNA molecule has how many base pairs? 34, in tRNA molecule, the anticodon arm possesses these. When out of these, you have to choose the correct option, that is how many base pairs are present in the anticodon arm. 35, T psi C arm in the tRNA molecule possesses the sequence. So out of these, again choose the option. 36, double helical structure model of DNA was proposed by. I'm doing it fast so that I can upload it on time. Okay. So 36 out of the four option correct choose kare aap log. 37 is DNA does not contain. Very easy question. 38, the sugar moiety present in. Again okay, yeah, so this was 37. I didn't skip. Okay. So 38, the sugar moiety present in DNA is. You have to choose the option. DNA rich in 80 base pairs have what kind of bonds? Very easy paper. 40, in DNA molecule, out of these four, choose the option. DNA rich in GC base pairs have how many bonds? 42, the fact that DNA bears the genetic information of an organism implies that. So what is the implication of the fact that DNA bears the genetic information? 43, the width or helical diameter of double stranded in B form DNA in nanometers is. 44, the number of base pair in single turn of B form DNA about the axis of the molecule is. 45, the distance spanned by one turn of B form DNA is. B form DNA. So what is the distance spanned by one turn of B form of DNA? 46, in DNA molecule, the thymine concentration, just forget this thing, just ignore this thing, whatever is written. So concentration is 30%. The guanosine concentration will be, agar they have given you thymine concentration, you have to guess how much guanosine is present there. 47, in a DNA molecule, the guanosine content is 40%. The adenine content will be. Very easy question. Use the Chargaff rule. 48, increased melting temperature of DNA duplex results from a high content of. Very easy question. 49, a synthetic nucleotide analog 4-hydroxy zy. Acha, 1 second. 4-hydroxy pyrazolopyrimidine. Pyrazolopyrimidine is used in the treatment of. This will be, I think, little bit difficult for you people. 50, a synthetic nucleotide analog used in the chemotherapy of cancer viral infection is. Again, a easy question. 51, histamine is formed from histidine by the enzyme histidine carboxylase in the presence of. Okay, you have to tell out of these what is present in the reaction when it is catalyzed by histidine decarboxylase. 52, infant Lee convo is ions due to lesser formation of gamma aminobutyric acid from glutamic acid is seen in deficiency of? Uh, you will have to search for this answer. I think many of you will not know. 53. Which of the following amino acids produce a vasoconstrictor on decarboxylation? Just forget this underscore X0002. Okay. 54. The degradation of RNA by pancreatic ribonuclease produces. 55. Intestinal nucleosides act on nucleosides and produce. Easy question. 56. In purine biosynthesis, carbon atoms at 4 and 5th position and at 7th position are contributed by. 57. N10 formyl and N5 N10 methyl tetrahydrofolate contributes purine carbon atoms at positions. This you have to. This I know. I mean, of course I know, but you guys will have to. I think you also know. This is from metabolism of nucleic acid. 58. In purine nucleus, nitrogen atom at one position is derived from. 59. The key substance in the synthesis of purine phosphoribosyl pyrophosphate is formed by. 60th question. In purine biosynthesis, ring closure in the molecule formyl glycinamide ribosyl 5-phosphate requires the cofactors. Very nice question. Ring closure of formidoimidazole carboxamide ribosyl 5-phosphate yields the first purine nucleotide, RUBP. 62. The cofactors required for synthesis of adenylosuccinate are. 63. Conversion of inosine IMP to xanthine monophosphate is catalyzed by, again from metabolism. 64. Phosphorylation of adenosine to AMP is catalyzed by. 65. The major determinant of the overall rate of de novo purine nucleotide biosynthesis is the concentration of. Pause the video and write your answer. 66. An enzyme which acts as allosteric regulator and sensitive to both concentration and purine nucleoside is. This is a very easy question, but it is much asked question, like most repeated question. PRPP, glutamyl aminotransferase, the first enzyme uniquely committed to purine synthesis, is feedback inhibited by, again a common question. For 68. Conversion of formyl glycinamide ribosyl 5-phosphate to form glycinamide ribosyl 5-phosphate is inhibited by. Okay, out of these you have to look for the drug or the inhibitor. 69. The biosynthesis of purine nucleotide, the MP feedback regulates. Other option, write your answer. 17th question. 6. Mercaptopurine inhibits the conversion of. Write your answer. From 71 you have questions till 70 you have questions from nucleic acid. Now the questions will start from your fats. The deficiency of both energy and protein causes. So this is a different question. So again you have to attempt the question, of course. And then 72. Kwashiorkor is characterized by. You have to write the characteristics of kwashiorkor. Now 73. A characteristic feature of kwashiorkor is. 74. Characteristic feature of marasmus is. I think it's going in the direction of what? Fat, proteins. 75. Obesity generally reflects excess intake of energy is often associated with the development of. Very logical question. 76. Atherosclerosis and coronary heart disease are associated with the diet. Easy question. 77. Cerebrovascular disease and hypertension is associated with. Easy question. 7. Normal range of total serum bilirubin is. It's a factual question. 79. Normal range of direct reacting conjugated serum bilirubin is. 80th. Normal range of indirect unconjugated bilirubin in serum is. See, it's not important that you should know all the answers, but I'm giving you difficult as well as easy questions just to give you a virtual feeling that in your exam you might get uncomfortable by seeing certain questions which you had never expected. Okay, so such questions I've also tried to incorporate. 81. Jaundice is visible when serum bilirubin exceeds. 82. An increase in serum unconjugated bilirubin occurs in. 83. One of the causes of hemolytic jaundice is. 84. Increased urobilinogen in urine and absence of bilirubin in the urine suggests. 85. Jaundice in which serum alanine transaminase and alkaline phosphatase are normal is. 86. Fecal stercobilinogen is increased in. 87. Fecal urobilinogen is increased in. A mixture of conjugated and unconjugated bilirubin is found in the circulation in. Hepatocellular jaundice as compared to pure obstructive type of jaundice is characterized by, I think 10 bouncer questions I've included for you guys. 90. An example of hydroxy fatty acid is. 91. An example of saturated fatty acid is. 92. The fatty acid is esterified with an alcohol of high molecular weight instead of glycerol. The resulting compound will be. 93. Fatty acid which is not synthesized in the body has to be supplied in the diet is. Essential fatty acid out of these is. The fatty acid present in cerebrocytes is. 96. Number of double bonds in arachidonic acid is. 97 in humans, a dietary essential fatty acid is, these I think you guys know. A lipid containing alcoholic amino residue is very easy. Cephalin consists of, again very easy. In mammals, a major part in adipose tissue is easy. Glycosphingolipids are a combination of easy. 102. The importance of phospholipid as constituents of membrane is because very easy. 103. In neutral fats, the unsaponificable matter includes. This is like I think it's a logical question. Okay. 104. Higher alcohol present in waxes is 105. Keratin consists of keratin, not keratin. Gangliosides are complex glycosphingolipids found in unsaturated fatty acid found in the cod liver oil containing 5 double bonds is, this is a factual question. Phospholipid acting as surfactant is 109. An oil which contains cyclic fatty acid and once used in the treatment of leprosy is 100. Unpleasant odors and taste in a fat rancidity can be delayed or prevented by the addition of 111. Gangliosides derived from glycosyl ceramide contain in addition one or more molecules of 112. Drying oil oxidizes spontaneously by atmospheric oxygen at ordinary temperature and forms a hard waterproof material is 113. Deterioration of food rancidity is due to presence of cholesterol, vitamin E, peroxidation of lipids, phenolic compound. With this, your question paper ends here, and thank you. Your feedback is appreciated. And after 1 hour, exactly after 1 hour, I'll upload the answer key. That is, it's 9:21 now, and I'll upload the answer key at 10:15. Okay. So thank you, everybody. I hope your test went well, and after this I'll give you like a virtual idea of as to how are we going to study, but I'll make the plan today and then I'll upload the video tomorrow. Okay, so for BARC Biosciences, I'll be giving you the entire plan till we'll study via my videos till 28th of March, and on 2nd of April you have your exam, and in between I'll take the test, and after some time I'll take daily tests, maybe 2 tests in a day. Okay, all I need is time to make this test because it requires effort, and I have to, even if I select questions from multiple sources, I have to look to the depth of the question, or can I include it, will it be useful for you or not. So that absolutely, this process takes time, and that all will happen, and you just study, and we'll meet after 1 hour that I'll upload the answer key. Okay, so thank you, everybody.